All right, guys, so we will cover Chapter 6, Part 2 today, brought to you from the inside of an Airstream. So, this part of the chapter really contemplates animal movement. We'll talk a little bit about plant movement at the end, but most of the science developed for tracking individual movement has been with animals. So, I'm guessing that many of you have been involved at some point in tracking individual movement of animals, if you have an interest in wildlife and are pursuing a master's. So, some of the different techniques that we see out there are mark recapture techniques. This is when individuals are marked with a sort of unique identifier and then either recited or recaptured again to gather data about their movement. Weather radar has been used to track really group movement of flocks of birds. We can track passerine movement at night. We can estimate the size of organisms moving through a weather radar and actually piece apart small birds, ducks, geese, and even insects. And then remote sensing of animals using transmitters that technology has advanced so much in recent decades and can easily be combined with other spatial information. And so we'll talk more about that. So each of these techniques have their trade-offs and benefits that ultimately influence the grain and the extent of the movement data and how it can be analyzed. So a little bit on mark and recapture or reciting studies. This is a brown-headed cowbird that I banded many years ago when I was doing my master's. And you can see the silver band on this bird is the USGS issued band number. And it's got a unique number that is assigned only to this individual. That data is housed at USGS's bird banding laboratory. And if this bird were to be recaptured by any other scientist netting birds, that information about the recapture would be sent back to me as the bander. But you see this bird also has a color combination on its legs, and that is also unique to this bird. So there's a second level of permission that a scientist can get to color band birds, which allows for reciting. As you can imagine, it would be hard to actually read the numbers on the aluminum band, and so those color bands allow for the bird to be recited and easily identified just with binoculars. So we've seen the use of dyes and powders, beads, streamers, fin, ear, and wing tags. We see leg bands and leg rings. When I first started in wildlife ecology, it was fairly common to do toe clipping on small mammals and create a unique combination of which toe had been clipped. There's been, as you can imagine, some ethical concerns around that. Marking individuals can be stressful and injurious, and so should be warranted. And marking assumes that at least some individuals, of course, are going to be recaptured or recited at a later date. Otherwise, it would not be warranted. And this kind of data can give us information on large-scale movements of populations, and population dynamics, structure, community composition, species longevity. Some of our most important records for how long bird species live come from recapture of individuals. For instance, some Carolina chickadees have been recaptured, showing that they can live up to 10 years. So it can also give us some fine scale movement data. For instance, the color-banded cowbird was an effort to mark its home range territory through resighting study. These are just some of the examples of mark and tracking devices that we see on different types of organisms. And then radio telemetry, a lot of the science of movement data has really stemmed from these advances in radio and satellite telemetry. It allows for collection of location data that can be measured in minutes, hours, days, or weekly intervals. Telemetry allows us to track movement across a broad range of spatial scales and even to collect really fine scale movement within a home range, or seasonal movements along a migration route across continents. So it's usually the cost and the size of satellite transmitters that limit their use. I'm sure most of you are familiar with handheld radio transmitters that where the data, the collection has to be done by hand by the scientist out in the field. And this is actually a picture of me placing a radio transmitter on a bird, on a swallowtail kite, before satellite transmitters were available in a small enough size to place. This is probably in the late 90s. So as satellite transmitters have gotten smaller, we actually see them being placed now on songbirds, bats, and other small vertebrates. And we're limited now, again, by the cost of those and also by the lifespan of the battery. It's been some really interesting science around stable isotope analysis. So what we have discerned is that various biochemical, biological, anthropogenic processes result in a distinct gradient and landscape signatures in the environment with the ratio of stable isotopes that then get reflected in the body tissues of individuals. So, for instance, with birds, if we're banding birds, we could remove a feather and study, use that feather to study the stable isotopes within it. So that indirect measures of movements that consider stable isotopes and other molecular techniques really have helped us expand the scope of movement studies. They could be used to uncover linkages between important ecosystems and assess the connectivity among migratory populations that breed and overwinter in different locations, giving us a signature to figure out where a bird overwinters without having to track it there. And we've seen this technology now used with birds and butterflies, sea turtles, fish, bats, and even whales have been subject to these types of studies. And as you can imagine, this type of analysis is going to give us a larger extent, but not as quite as a fine grain. We're not going to look at small, detailed movement data with stable isotope analysis. So when we are using telemetry and now more and more satellite telemetry to track animal movement, the interval of our data collection is going to ultimately affect the grain and extent of our data. So there are really issues both with sampling that data and with oversampling position data. We can program our devices to collect on the minute, on the hour, or only once a day, and you can imagine the resolution of the data you get on those different on those different temporal scales. So satellite data now actually results with more issues in oversampling than undersampling, and there have been some statistical approaches developed that have been used to assess the degree of autocorrelation among movement steps to determine what scale the movement might be adding to our understanding of how that individual is actually interacting with patch structure. A lot of our analyses do assume some independence of movement in the measurements that are taken. So it's important for us to correct for that. And you can see in the figure just the different resolution there and that effect of temporal scale on what the movement pathway is going to look like for an organism. This table just describes some of the parameters used most commonly to describe animal movement pathways, and so I will not read this to you, but you guys can read through it. So fractal analysis of animal pathways is a way for us to understand the effect of habitat structure on the movement of individuals. It provides a way to quantify the spatial complexity of a movement by assessing how the structure of the pathway varies as a function of scale. So it can be used to understand movement pathways within a particular domain of scale or even scale-dependent shifts. In this particular study, they looked at 3 different darkling species of beetles with different movement rates that actually showed a similar movement pattern, meaning they were responding similarly to patch structure. This is an example that the book gave of being able to identify a shift in movement based on different domains of scale. So we might see a different movement pattern within the scale of a foraging range or a foraging bout, and then a real shift in that as we scale up and perhaps organisms are orienting themselves around something else in the patch or the landscape. So there are several different ways we try to model or anticipate animal movement to understand it better. The most simple of these are just simple random walks that assumes that movements are uncorrelated, independent movements, unbiased, direction is completely random. And then we see increasing complexity in the way that we model these movements. So we take that a step further with correlated random walks, which really acknowledges that once an animal is moving in a certain direction, that it's likely to continue moving in that direction. Biased correlated random walks take it a step further by accounting for the fact that we've got directional movement and that might be biased towards a certain habitat or landscape feature, and that we can predict for that if we have enough knowledge of the organism. Levee walks or flights. So over longer spatial and temporal scales, we know we often see movement that involves sort of concentrated searching separated by longer movement between patches. So that pattern of movement has really been found to be ubiquitous among lots of different types of animals. And we know that that behavior is probably adaptive for being able to exploit resources in a heterogeneous landscape. So it's important to note that models that seek to understand movement pattern and relate them to landscape features of resource availability may change as we change the spatial and temporal scale of our observations. So spatially explicit simulation models help us understand how individuals interact with a spatially complex landscape. So recall in chapter 5, we talked a good bit about neutral landscape models and how that would affect movement through corridors. So we can take this analysis a step further by applying some of the rules of movement of an organism into neutral landscape models and predict, based on our knowledge of certain species, how far, how fast, what direction, and what habitats organisms are likely to move through. And that really allows us to explore the relationship between an individual movement pattern based on our understanding of how organisms perceive the environment and landscape structure. So let's talk a little bit about space use analysis and home range analysis. We talked in the last lecture about what a home range is. So home range is the area around the home of an individual where most of the normal activities of gathering food, mating, caring for its young take place. As you'll remember, we talked about the fact that the home range and the territory or the defended area are not always the same for given individual or species. Home range is often larger than the territory or the defended area. So an analysis of space use within a home range can reveal habitat preferences. We can discern and see the habitats that are being selected for. Those are habitats being used disproportionately to their abundance within the landscape, and conversely, we might see certain habitats that are avoided. So quantifying the size and the distribution of home ranges can help us estimate the carrying capacity of a certain geography for individuals. It can help us try to extrapolate or understand population size, assuming that home ranges are relatively static. In this map, we see the home range that was tracked via handheld telemetry by a graduate student of mine in the lower Black River. And this is a swallowtail kite and its movements over the course of, I guess, about 6 weeks. This bird was tracked. So a few different methods of home range estimation that are discussed in the book. The first, the minimum convex polygon, MCP, is the smallest area that contains all of the recorded locations for a particular individual. And this is a bit of a dated technique that we don't see used too much. It's generally considered really sensitive to the sampling protocol that a researcher applies. So overly sensitive to sample size, methods, duration, and we're going to get really different results with our MCP depending on that sampling regime. So it's something that you're probably going to come across in the literature at some point, but less likely to actually see applied today. The more dependable and most frequently used now is the kernel density estimator, and that's what we're seeing in the figure on the right. In KDEs, the home range is viewed as a three-dimensional surface, and the height corresponds with the amount of time that the animal spent in a given area within the area of analysis. So KDE is a better predictor of actual home range than MCPs. A KDE may only require 30 to 50 points, while an MCP would require 100 to 300 really to reach the same amount of accuracy. And intuitively, a KDE's accuracy is going to increase to a certain point with more points sampled. But we have to be careful, even with kernel density estimators, to understand the organisms that we're studying. We have to account for the time spent in resting locations that can bias our data, and also organisms with dynamic home ranges. And there are some ways that we can correct for some of these behaviors once we have our sampling done. And just a few notes on the study of individual plant movement. As you can imagine, this is not an easy task to study plant dispersal. There are, we know, a wide variety of dispersal mechanisms that aid in plant movement away from natal territories. So movement of plants and dispersal of plants across the landscape. And understanding those mechanisms of plant dispersal is really important for us to understand spatial distribution, dynamics, the spread of plant species across the landscape. As you can imagine, this is really important for understanding and anticipating the spread of invasive plant species. We also know that microsites within the landscape, so those sites that are ideal for plants to be able to germinate for successful recruitment, are patchily distributed, and there are some methods that scientists have used to track individual propagules to see where they end up. That is a really labor-intensive effort. We can also draw some inferences about dispersal of plants from the number of plant propagules that end up in seed traps in a given location, and so this is a method that we've seen applied a good bit. And then botanists have also used some mark recapture techniques for plants, but for most plants, seed trapping, modeling dispersal, and even the use of molecular markers gives us a better understanding of dispersal of plants. And these are techniques I'm sure that we'll see new innovations and new approaches in years to come.